Welcome to the first episode of the Pittcon Podcast. In this episode, we speak to the recipient of the Analytical Chemistry Award for 2023, Neil Kelleher.
I'm Neil Kelleher. I'm the director of the Chemistry of Life Processes Institute at Northwestern Proteomics at Northwestern University in Chicago and Evanston, Illinois, right in the middle of the United States. And I do top-down proteomics, which we'll talk about.
And what first attracted you to that field?
So I was first pulled into science when I was 15 years old. And I worked at a company called Weyerhaeuser, a tree and paper company out in Seattle, Washington, in the very Northwest. And there I discovered analytical chemistry. Some scientist gave me, I was a high school student, and he gave me a tin of sulfur, some yellow compound, and said, you have 4 months to identify what this is. And I learned spectroscopy, infrared spectroscopy, and I was 15 years old. So that just led me into a career in biological mass spectrometry.
Yeah, what is proteomics?
Yeah, what is proteomics? That's a great question. It's a sprawling field that has to do with protein-level biology. And so it's the study of proteins, and I guess the word is probably the best way to answer the question, what is proteomics? If genomics is the study of all the genes in an organism or an ecosystem, then proteomics is the study of all the proteins collectively, which has really diverse features, and we'll talk more about it.
Yeah, and what is the difference in understanding that proteomics provides compared to genomics? So what can we learn from proteomics that genomics can't teach us?
Yeah, so most people are familiar with the so-called central dogma, or DNA gives rise through RNA molecules to proteins. And the proteins are the worker bees in your bodies. If you point to anywhere on your body, you're pointing to proteins. And so it's thought that proteins are closer to our biology and disease traits, closer to our complex traits. And some of the parts of your body, like your eyeball, it's all made up of proteins, and they don't get renewed throughout your life. So we really need to understand proteins to understand holistically human biology.
Yeah, that's fascinating. And how does Fourier transform-based mass spectrometry impact the interpretation of measurements in proteomic applications?
So Fourier transform mass spectrometry is sort of like the Porsche of mass spectrometers, or has been, ultra-high resolution. And it's FTMS, which breaks down into various types of Fourier derivatives of mass spectrometry. But the famous ones are, one that uses a superconducting magnet, like an MRI in a hospital. So we use that style. And then there's Orbitrap style FTMS. And they've had a big impact in protein measurements in proteomics.
Long considered a specialist technology for physicians and chemists, how important is mass spectrometry in biological research?
Yeah, FTMS, or Fourier transform mass spectrometry, is playing an increasing role in biological research, both metabolomics, proteomics, study of the proteins, imaging biological tissues for both those things, lipids, energy metabolites, proteins of ever larger size, and that means also capturing proteoforms, which we'll define in a moment.
So what is top-down proteomics?
Top-down proteomics is it's a philosophy that we came up with at Cornell to first measure the molecular weight, or the composition of the whole protein, and then controllably degrade it into pieces. Don't create the pieces first and then try to build up the protein bottom up. So it's a measurement philosophy, it's a measurement approach, top-down philosophy, and it has as its benefit determining proteins with complete molecular composition. So top-down proteomics, as a last point, it's got the benefit of determining proteins with complete molecular specificity.
Yeah, brilliant. You've answered a little bit of the next question now, but what are the advantages and disadvantages of top-down proteomics?
Yeah, so the advantages of that top-down philosophy of molecular analysis now applied to proteomics, so top-down proteomics. The advantages are to be absolutely precise about molecular analysis of proteins. The disadvantage is that the implementation of it still is lacking, and we need about 100-fold boost in that ability to capture all the proteoforms, all the different forms of protein molecules in a complex sample like human blood or tissue from any part of the body or other biological model systems.
How is it used in research, this top-down approach?
Top-down proteomics, it's used like other proteomics tools in both basic biomedical research and clinical translation. And I would say for that last part, there are some real sources of excitement of the concept of implementing proteomics with complete molecular specificity, so top-down proteomics, which determines proteoforms. If you do that in cohorts of patients, 100, 200 patients, you will detect strong biological signals as biomarkers. And that is something that the world desperately needs, to detect disease earlier so that you can involve yourself clinically and try and fix what is wrong with people. You've got to detect it earlier. And top-down proteomics is a major part of that solution.
Yeah, definitely. You've mentioned this word a few times. I was just wondering what is actually meant by the term proteoform.
So proteoform, that's a great question. And for me and the people in this consortium for top-down proteomics, it's not just a word, it's a movement. And for that, let's understand what we mean. We mean that a level 1 proteoform is when you know the complete atom composition of a protein molecule. And that comes from one of the 20,300 human genes. So those would be human proteoforms. And we're interested in creating the world's atlas, the definitive reference set of proteoforms in human biology. And that is called the Human Proteoform Project.
Wow, that's really interesting. And how does your current work aim to bridge the gap between genotypes and phenotypes?
That's a great question. So that is really, in biology, there's a widely understood gap between the genes that encode the molecules that give rise to life and the phenotypes. So genotype to phenotype gap. Said plainly, it's genes to complex traits, often disease traits if you're talking about humans or model systems. So that gap, there are many omics technologies, exposomics, metabolomics, transcriptomics, proteomics, but there's a thought that proteins really should be a strong connector, but they aren't right now. And so to close that gap, applying top-down proteomics to determine proteoforms will connect genes to phenotypes for both basic biomedical research and especially clinical translations. Clinical research, deploying that in the clinic, it will take a few more years because proteoform measurement is a relatively small area but growing quickly.
I was just wondering as well, what impact has the field of proteomics had on a study of Covid19 and the causative agent SARS-CoV-2?
Yeah, so the pandemic, we've all just been through a moment. It's been a hot moment. Oh boy, 3 years. And that's a classic case where to know, okay, it's an RNA virus, but the proteins and the envelopes, we actually have a full catalog of the proteins that are present. And because of the focus of biomedical research, there's a large amount of proteoforms known from SARS-CoV-2. And that's one way that we've understood, okay, what is the virus? Where can we target drugs against the virus? So that is the beautiful case of proteomics writ large as a field, impacting positively biomedical research. But there's another area, and the clinical side, when you talk about serology and immunoglobulins, antibodies as drugs, proteomics has had a major impact in both the detection and treatment of COVID-19.
What does it mean to you to be this year's recipient of the Pittsburgh Conference Achievement Award?
For me to get this Pittcon Achievement Award, I just can't be more excited. I am elated. I was like a cat with the claws up on the ceiling. When I got the news a couple months ago from Brian and the award committee, I was just absolutely floored. and I've been excited ever since. He tells this great story of the day that I got the news that I was the Pittcon Analytical Achievement Award winner. I was having 2 students defend their thesis at Northwestern University, and that just put it over the top. You know, I had their families in town, my group of about 45 people at Northwestern. It was just like, it was the best day in well over 3 years in the pandemic.
So you had a nice little celebration.
I had a lovely celebration, and there could have been champagne involved.
Could have been.
Yeah.
And what are you working on right now that you're particularly excited about?
Right, so I'm particularly excited about the prospects of reaching a tipping point on the Human Proteoform project. So it's the equivalent of the Genome Project, but for proteins. Let's sequence, map, define the human proteome like we did the genome 2 decades ago, and it'll take another $2 to $3 billion or euros, take your pick. It's a worldwide project to really clarify the human proteome, and that means protein level biology. Just what are the molecules? How many of them are there that are created from the 20,300 human genes? And that is really exciting. And there has been, in the private sector, a major move in the last 3 years. Proteomics companies have gone public. They have had major investments in large venture capital and institutional investors. And so now we need the governments of the world to take a step in and say, those companies will be advantaged tremendously if they have the set of reference proteoforms in human biology.